So you may have realized that I'm not Astrid. When we got here and we saw her name in the program, I told her, you have to give this talk. And Astrid just looked at me and said, no. And you need to know that Astrid is a former German champion in judo. So I didn't dare to argue with her about that, so I got to do it with her. I'm sure almost everybody in this audience has come to a stage where you had a crystal that wasn't suitable for diffraction studies and needed further improvement. And when you look what you can do about optimizing a crystal for diffraction, there's only 3 general things that are possible. Number 1 is you optimize your crystallization experiment itself. Number 2 is you optimize your protein that you put in the crystallization experiment, or as a salvage strategy, you optimize or you try to rescue the crystal. I'm not going to talk about that today. I'm not going to talk about that today. Neither I'm going to talk about how to modify the existing protein. If anybody's interested in that, you're very welcome to stop by our booth that is just outside this lecture hall. What I'm going to talk about today is what can we do about making better protein more suitable for protein crystallization. And the major topic there will be the mode of production. Among crystallographers, it's like totally accepted that the major mode for production of proteins is a recombinant expression in E. coli. It's a wonderful method, everybody is using it, and the question is, why should one change anything there? And there's one fundamental reason. When E. coli produces proteins at the ribosome, and the proteins leave the ribosome, E. coli is not able to do anything with those proteins anymore. There is no active chaperone system to filter proteins such as known in the eukaryotic cells. And there is no machinery for post-translational modifications with the protein, which is very important for most of the interesting proteins. I'm just going to describe some of the symptoms that some of you may have seen with the protein preparations. Yeah? Whenever you have insoluble protein that is only expressed in inclusion bodies, or when you have a protein that forms aggregates, which you can easily see when you take your protein preparation and spin it down in a centrifuge for some time, and you get decreasing concentration every time you do it. This tells you your protein is aggregating. Or if you have trouble concentrating your protein to a concentration around 10 mg per mil, sure, everybody, or almost everybody has seen that. You have one of those centrifuge devices for concentration with the cut-off membrane, and you centrifuge forever and ever and nothing happens. Or you have your protein is not stable, you see fast degradation, and very likely this is due to incorrect folding of the protein, of your protein preparation. Or second set of symptoms, kind of similar to the above. If you have only low expression yields, again, unstable, fast degradation. If you have trouble with the biological activity of your protein, or again, you have low solubility, it might be due to the lack of post-translational modifications. One needs to know that about 50% of all human proteins are glycosylated. So you're missing out on a lot of space here. And the major question, of course, now is, if E. coli delivers a bad protein, what can we do? And this is a really difficult trade-off, what to do. It's like staying with E. coli and doing a whole set of optimization experiments with the original protein preparation. So there's plenty of opportunities what you can do, or you go and change your mode of production, which of course is a lot of work because you have to start from scratch again. And I'm going to show you 2 methods today that entirely change the mode of protein production for crystallization that are not actually a hell of a lot of work to do them. And the first one is Lexi. May I introduce Lexi? Lexi is this little parasite here, Leishmania tarantula. It's a unicellular organism, has a little flagella at the end, and moves lives in liquid medium. Originally, it's a parasite of this gecko here. And we at Jena Bioscience, we have transformed this Lexi, this Leishmania tarantula cells, into an expression system. They're safe to work with. They have a biosafety level 1 as E. coli. They're easy to manipulate in terms of molecular biology. You can use E. coli shuttle vectors, so all the molecular biology is done in E. coli. Just the final step of transfection is in the Lexi. They grow in inexpensive media, 26 degrees, and they achieve expression yields so far up to 300 mg per liter of culture. The good thing about them, they have the fully eukaryotic folding and modification machinery. So they have all the chaperones, so no inclusion bodies ever, and they have the full range of the post-translational modifications such as phosphorylation, disulfide formation, granulation, lipidation, glycosylation, everything. And for the glycosylation, there's an interesting fact that we discovered when we compared erythropoietin that was expressed in Lexi. You see here that this is almost a sharp band, whereas the comparison, the echo produced in mammalian cells is a smear. So the smear here is due to that from the mammalian cells, they deliver a large variety of glycan species on the protein, whereas from the Lexi system, only a single species occurs. This was verified by sequencing, glycan sequencing using mass spectrometry. And of course, for crystallization, having a single species is always better than having a mixture of species. The system comes in several configurations. You can do it inducible or non-inducible. You can have the protein expressed in the cell or the protein secreted to the medium, so anything you like, it's actually there. Now the major question, of course, does it work for X-ray crystallography? And it was just a paper published a few weeks ago where human copper zinc superoxide dismutase that was expressed in Lexi was crystallized in the structure solved. So what you do, you subclone your gene of interest into such a vector. You transfect and select. These things grow on agar plates, similar to E. coli. This is fused to a fluorescent reporter protein, so you can immediately also screen by color. The red color tells you there's overexpression of your protein of interest. Here you see before induction, there is nothing. You see post-induction, there's a strong band. This is simple his tag purification, so you end up with recombinant protein. The whole process takes about 4 to 6 weeks. It's not 4 to 6 weeks working full time on it. There's a lot of incubation time and growth time, but in general, to start with the first step and end up with a purified protein, we're talking about roughly 4 to 6 weeks. The protein was crystallized and the structure was solved and was recently published, as I said before. And 4 to 6 weeks is pretty good, but isn't there anything shorter? Because we don't want to go back to the wet lab all the time. We want to change a protein. And yes, there is something quicker or something shorter. The method was published last year. It was a title cover in Nature Biotechnology. It's the in vitro version of Lexi. So what we basically do, we grow Lexi cells under certain conditions. We harvest those cells. We extract their translation containing extracts, freeze those, prepare those, and those can be used for in vitro cell-free expression of proteins. So there's no cell culturing necessary, and the yields reach about 0.3 mg per mil, and this is done within 2 hours. And this gel here just shows you a few examples of proteins that were done. I don't think it's worth to go through here in detail, so it's very fast. So how does it work? Similar workflow than the last one I showed you. You need to prepare template DNA. At the moment, this must be done by a subcloning step, a ligation step into the appropriate vector. We are working on a method where you can use crude PCR product just from your gene of interest, but this is not available yet. It's going to be within the next month. Then you inoculate your cell extract on the bench. You leave that for 2 hours, then you detect your protein and you purify it. However, protein resulting from this system has not been shown to be suitable for crystallization and for a high resolution structure. Maybe anybody with you would be interested in doing that. Here's the offer. Anybody who sends a preprint of a structure with an accepted paper to this email address that shows the structure from the in vitro Lexi derived protein is going to receive 2,500 euros in consumables from Jena Bioscience. So just take it home as a take-home message and send me an email whenever you're there. To sum this all up, there are 2 straightforward choices for skipping E. coli. On the left-hand side, the traditional in vivo expression system. We have programmable Lexi cells here, S1 cells, expressing EGFP here. They reach up to 300 mg per ml of culture, and they're already proven for structure determination, a crystal structure of SLD1, and an NMR structure of GFP. The advantages, they eliminate all the shortcomings of E. coli. So they do have the folding system, they do have the post-translational modification system, everything that you want. They're very easy to handle. You don't need any special equipment for cell cultures. The disadvantage certainly is you have to go back to the wet lab. You have to do all the cloning, transfection, selection, and purification business again. So we're talking about 4 to 6 weeks here in total. The second one would be the in vitro expression, cell-free. In this case, we have a programmable cell extract. Everything is done in vitro. The yields reach up to 0.3 mg per ml of cell extract. So don't mix this up. This is 300 mg per liter of culture, and this is 0.3 mg per milliliter of cell extract. So this is different. Cell extract is much more expensive than liquid medium. It is not yet proven for structure determination. The advantages, of course, are the same. Eliminates the E. coli shortcomings, easy to handle, very fast results, 2 hours, and you know whether you're going to have a protein or not. The disadvantage, of course, is the lower yields and the associated more expensive consumables. But if you don't want to make a lot of protein, just enough for, say, a rough screening to see if that one might be the method of choice, it's actually a suitable system. These are the people who have contributed to this work. Kirill Alexandrov and his group from the University of Brisbane. Australia, Wolf Longenfeld group from Max Planck in Dortmund. Peter Weyer, who solved the structure of the NMR structure of GFP. Those days in Halle, now I think he's in Essen. And this is Astrid. This is Reinhard, who has been with the Lexi system for 12 years. When we started exploiting this technology and developing this technology. And this is Thomas, who's also around. And as I said before, anybody who has some more questions or wants to talk about other things for crystal optimization, you're welcome to stop by our booth outside. Thank you very much for your attention.
Thank you for this clear presentation. Questions? There.
Is it possible to apply those systems also for the expression of mammalian membrane proteins, for example? I mean, what should Lexi do with the membrane protein? Can it introduce it into its membrane?
No, it can't. It can't. So it can be used.
It can be used for expression of any protein, but the same principles that apply for membrane protein expression in any other host do apply for Lexi as well. We're working on a project, a proof of principle for GPCRs, but it has just been initiated, so I can't tell you any results about that. But you will definitely need detergents to stabilize the protein.
There was a second one. Mike?
One question, according to the yield of the cell-free expression system, could you increase that by mixing, for example, use the translational apparatus of E. coli, and then the post-translational modification of the leishmanian? maybe the translational apparatus is more powerful from E. coli. You understand?
I don't know. The preparation of the cell extract is tricky. So, and if you start mixing that with something else, I severely doubt it's going to work. What I can tell you though is we did a systematic comparison of the yields of the cell-free Lexi versus the established system. There's a cell-free E. coli system commercially available. There's wheat germ system available, and there's some rabbit cells that's also available. And those systems all achieve yields in between 0.1 and 0.2 mg per mil. In the Lexi system, we reached 2.3, so that tells me that the yields there are higher. And I don't know if mixing any of those components would make an improvement. I have no idea, though we've never done this experiment.
The last question.
Hi, how does the price compare to HEC cultivation, for example? The culture meter, how is the price compared to the radial cell culture? And what kind of antibiotics do you use for selecting?
Yeah. So the media are cheaper than for mammalian cultures. I don't have the exact numbers here. I would say about a factor of 2 or 3 cheaper. They're more expensive than LB though, that you use for E. coli. And there are 4 antibiotics available that we use. The standard one is NTC, norcerotricin. And you can also work with bleomycin, and there are 2 others, I don't know at the moment. But there are 4 antibiotics that work. So there are 4 resistance in the system.
Do you need serum for the cultivation?